Hello, my name is Katie Parzych and I'm a graduate student in the laboratory of Dr. Daniel Kliansky at the University of Michigan. I think most of us would agree that recycling is a pretty good thing. We can collect our waste and unnecessary metals, papers, and plastics, take them to a central recycling center, and they are broken down into small pieces, and these pieces can be used to make new things. It prevents a lot of waste from building up in our homes and in landfills. But would it surprise you to know that each and every one of the cells in our body can do essentially the same thing? In cells, this process is called autophagy, which roughly translates to self-eating. Now, like I said, all of our cells do this, but human cells can be difficult to work with, and we don't have very well-developed tools for studying autophagy in human or mammalian cells. Fortunately for us, the process of autophagy is very similar between human cells and yeast cells, the same sort of yeast that is used in beer and bread. And we have much better developed tools for studying autophagy in yeast, so we use this as a model. In every yeast cell, there is a recycling center called a vacuole. The cell identifies the pieces to be recycled and wraps them up in a sort of garbage bag that we call an autophagosome. This autophagosome takes its cargo to the vacuole where it unloads the cargo and it is then broken down into its smallest pieces, and these pieces are then sent back out into the cell where they can be used to build new parts. Over time, parts of a cell can break down through use and wear. The cell will selectively recognize these broken pieces, package them in autophagosomes, and the autophagosomes are taken to the vacuole where they are destroyed, and the parts can be reused. Because the cell is carefully selecting the pieces to be degraded, this is called selective autophagy and serves a maintenance or housekeeping function. This process, though, can also be used in times of crisis. If a cell senses that it is starving, instead of targeting specific parts, it will wrap up random cargo that can then be taken to the vacuole for degradation, and those pieces are used as a nutrient source until conditions improve. Because the cell is not carefully selecting its cargo, this is called non-selective autophagy and is necessary for survival in starvation conditions. As with anything, balance is key. In our own homes, if we don't take out the trash and recycling often enough, we can find ourselves in an unhealthy and potentially dangerous situation. This is also true in cells. In cells where there is too little autophagy and they cannot take out the trash and broken parts, this can contribute to a variety of human diseases, including Alzheimer's, Parkinson's, cancer, and many other diseases. However, the flip side is also a concern. If you got carried away taking out the trash, you might start to throw out your chairs, your windows, your doors, and pretty soon the whole building's coming down around you. Now, I'll admit this is an extreme example, but in cells, this is a real concern. In cells that can't turn off autophagy or where autophagy is too high, cells can actually eat themselves to death. Cells must constantly be surveying their internal and external environments to determine the appropriate level of autophagy to maintain a healthy, clean, balanced state. There are several ways in which cells can change the level of autophagy. One is through altering the capacity. Cells can change the number and size of the autophagosomes used to transport cargo to change the overall capacity of cargo that can be taken to the vacuole. Another way, and the one that I'm most interested in, is how do cells turn autophagy on or off in the first place? Cells are constantly sensing their external environment to determine whether or not they are in a starvation situation. When a cell is in an environment where nutrients such as amino acids, nitrogen, oxygen, and sugars are in abundance, the cell can sense these nutrients and send a signal to the autophagy machinery to keep it low or completely off. However, when any one of these is in short supply, a cell can sense this change and transmit a signal to the autophagy machinery to turn it on. We know several of the key regulators that transmit these various signals to change the level of autophagy, but the puzzle is far from complete. And this is where I come in. My project in the lab is dedicated to identifying and understanding additional regulators that transmit these signals. The hope is that by gaining a more complete picture of how cells control autophagy, others can develop new and better ways to adjust the level of autophagy in diseases where it is too low or too high to restore cells to a healthy, balanced state and provide better treatments for a number of human diseases.